So our TTM program are models that we offer off the shelf with full licensing. So what that means is you call us up. If you want 100 for a study, we can give you 100, hopefully next week. That means you don't have to have some sort of colony that you maintain at your facility, that you have to maintain this huge colony. Even if you only want 100 once, you would need to have enough breeding that you could get 100, and it takes a while. We have full licensing, so if you're a pharma company, you don't need to call up DuPont and get a license for something. You don't need to call up whoever did the gene. We take care of all of that, and so that's included in the price of your mouse. There's conditions of use, which basically say we retain title, you can't breed them, a couple other things, but really it gives you full use license, and so that's why it's really popular among pharma. And we look for models that complement our current portfolio of models, because certainly the sum of the parts is worth more than each individual model. And so these are models that we in-license from the investigator, from the tech transfer office, and we pay a royalty. So you give them to us, we bear all the costs for setting up that colony, and then we pay you when we start selling them, basically. But there really are some significant startup costs, because they're generally pretty large colonies. It takes some time, so we really need to have a pretty good idea that there's a market for this model before we would take that risk. So this is our current portfolio of models. Short-term carcinogenicity, I don't know how familiar you are with the drug discovery development process. Certainly for any drug, before it goes into clinical trials, they have to do safety testing, see if it induces cancer in humans. The old way to do this was a 2-year bioassay, which is basically a lifetime assay. That takes a long time. Time is money for pharma, so if they can do something in a shorter amount of time, they'll do it. We have a number of models, the P53 heterozygous knockout, the RAS-H2 model, the TGAC model, which don't get cancer or tumors spontaneously, but which, when carcinogens are applied, have reduced latency to develop tumors. And so you can do this assay in 6 months. So that's a really great research tool that you can see applies to every single drug. And so you see there's a really broad application in a broad market. The tumor biology models, these tend to be a little more primarily academic. ADME models, we have a number of drug transporter knockouts. And so certainly drug companies, again, this applies to every drug. If they see it's being transported by BCRP in an in vitro assay, they can back that up or do some methodological studies in mouse models, in vivo, before they go into humans with it. We have a large portfolio of immunology models. Again, that's more a primarily academic market, although things like the RAG2 is another one of those pretty popular TTMs that applies to academics and to pharma. And you can see how it applies to people doing a lot of different types of research. Disease models are really big. We like to say that anything that's a baby boomer disease, we're probably going to have a pretty good market for, because that's where the pharma dollars are. So if you arthritis, any kind of cardiovascular disease, metabolic disease, these are really big things now that there's a lot of money being put into. And so if a company identifies a good mouse model, they could buy $500 a month to screen all of their early lead compounds. And then certainly neurodegeneration, Alzheimer's, another one of those baby boomer diseases, huge, huge. So Alzheimer's models are something we're definitely looking at.